So I'm going to continue and talk some more about the different mechanisms by which a bacteria can become resistant on a molecular level. So the first one was drug inactivation. That's pretty straightforward. You clip it, boom, dead. Nothing. The antibiotic can't do anything. A second mechanism, though, is target modification. And that is shown here. I lost my pointer. So a simple example of how this works is shown here. If you have a target protein, the antibiotic will fit to some site on that protein, and they have to fit together like a lock and key. If the cell is able to modify the target such that the antibiotic can no longer bind to it, it becomes resistant. So this is extremely common, for example, in the ribosome, where a mutation in the small subunit of the ribosome, one single mutation, can make the cells resistant to the antibiotic streptomycin. So it doesn't need a lot of changes sometimes. It could be very simple with one change. Another example I'm going to show you is a little more complicated. This is vancomycin, and I didn't talk about the mechanism of vancomycin yet. It's used to treat MRSA, the methicillin-resistant Staph aureus. It's a gram-positive bacteria, and this is a much more complicated structure. And what happens, I showed this slide last week when I was talking about penicillins, which shows the peptidoglycan here, and these penicillin binding proteins which create cross-links in the peptides coming off the glycan strand. And you need that for strength and keeping the cell wall intact. What vancomycin does is shown here. It binds to the peptidoglycan, but only of gram-positive bacteria. It binds and it prevents, it covers up this amino part of the peptidoglycan, and this cross-linking enzyme can't fit anymore, basically. And so eventually the cell falls apart, or the cell wall falls apart, and the cell's going to die. What vancomycin resistance is, though, is very clever. What has happened here, and I don't know how well you can see this, if you look at that little red ball on the end here, that's an alanine in the original structure. What resistant cells have done is exchange that for D-lactate. So they have an enzyme, they have evolved a new enzyme that puts lactate in there instead of the alanine. The cross-linking enzyme can still work. That's fine. But vancomycin can't bind. Okay, so that's another mechanism. You change the target that the antibiotic binds to. And that's seen in lots of different ways, or different antibiotics. Another type is something called an EFFLUX pump. And these are two examples of EFFLUX pumps. There's many different types. This is showing the membrane, the inner membrane of the cell. And there are proteins that go through the membrane, or protein complexes, in this case, they go through both the inner membrane and the outer membrane. And what they do is they pump out the antibiotic. So it's not so much that the antibiotic doesn't work. The antibiotic comes in, but then it gets shot right back out again. So it never, for example, finds its target. So one example, fluoroquinolones, as we talked about, the concentration of fluoroquinolones in the cell never gets high enough for it to effectively stop DNA replication. And there are many of these many different types of EFFLUX pumps. Two types, that one is the MATE family and the other is the R&D family. And I'll mention those a little later. Another thing these often do is, I should mention what this sodium here and this proton is here, is, or it's usually coupled to influx of another molecule, a sodium atom or a hydrogen. In the case of the R&D family, the reason these are really scary is that there are many drugs that can be pumped out. Some are specific for one or two drugs, but others are very generalized and can pump out a lot of different antibiotics. Okay, another example of a way in which a cell can become resistant is bypass. So I showed you this picture before, where you have these two drugs, the sulfonamides and the trimethoprim, which both turn PABA into folic acid. And folic acid is needed for a lot of things in the cell. The way in which cells become resistant to this, there are multiple ways. One way is by overproducing PABA. So the cell has evolved enzymes that it is making extra PABA, and then that overwhelms this inhibition. If you have a lot of the input, a little bit of it gets through, which is enough for the cell to grow. The other way is, some bacteria have evolved another way of making folate that just skips these enzymes entirely. They just go another pathway. Okay, so those are the ways in which cells can gain abilities to become resistant. But then there's something called natural resistance. I put that in quotes. Some bacteria simply are naturally resistant to some antibiotics. For example, many gram-negative bacteria are resistant to drugs that target gram-positives simply because they're less permeable. The drugs can't get in. They can't get through the lipopolysaccharide and the outer membrane. But in addition, some states of bacteria are more resistant than others. For example, cells that are not growing are generally more resistant to many antibiotics. I mentioned the penicillins in particular that have a very different effect on non-growing cells because they're not making new peptidoglycan. But also in general, they tend to be more resistant while they're non-growing. So if you remove the antibiotic, they could start growing again. The other example of a natural state is a biofilm. And cells in biofilms can be much more resistant to antibiotics. I'm going to explain what a biofilm is. So this is a picture, an electron micrograph of a biofilm that was taken from a catheter. So this is Staph aureus that is growing on the surface of that catheter. Most bacteria like to live in communities on surfaces. Almost all bacteria do this. And they do this in a very particular way. It's not just a random event. What they do is shown here. Bacteria will first be hanging around near a surface. They then attach to the surface in a specific way. They then begin producing colonies of bacteria, so more and more bacteria. And at the same time, they start producing what's called EPS, or extra polysaccharide. And this is a, how to describe this, a gel-like structure around them. They then will start growing up like this and forming these megastructures. And even bigger here. And eventually they will get so big that some of the bacteria will leave and they will move on and hopefully make their own biofilm someday. This shows also pictures of the surface. Now where you've seen a biofilm, I know you've seen a biofilm, your teeth in the morning, you're growing a biofilm, and that's why you brush your teeth. Rocks in a river tend to have a biofilm on them. Almost anywhere where they cause problem for us are things like catheters and things that come in contact with the body. So, why are they resistant? Why would a bacterial cell in a biofilm be more resistant to antibiotics in general? Can you think of any reasons? And this is a little bit of a trick question in that there are many reasons. So you can discuss for a minute. So there are many reasons, and we don't know all the reasons yet. I mean, people are studying this a lot. There are also different metabolic states for these cells. So the cells that are near the surface or deeper in the biofilm, they don't get much air, they don't get much oxygen, and they also might be limited for nutrients. And so those cells may be more resistant because of these factors. Then we have protection from the extracytoplasmic polysaccharides and bacteria near the surface. I wrote that poorly. Near the surface they're growing on might be more difficult to reach, that the antibiotic can't penetrate that far. Okay, so where does all this resistance come from? The natural resistance is kind of easy to understand, but where are all these genes coming from? What's happening here? So I'm going to talk a little about evolution and gene transfer. So how does antibiotic resistance arise? Well, like most things, in basic evolution, you have genetic variation. So here we have a number of cells. They're different in some way. If you apply a selection, which could be an antibiotic, you will select for ones that can grow. So the ones that are the most red or orange here grow the most. They survive the antibiotic. They are who wins, so to speak. And that's how resistance arises in a simple way. Where does this genetic variation come from? In one way it arises is through mutations. Mutations can arise from damage to DNA and from mistakes during the replication. So the replication is very accurate, but occasionally it puts in the wrong base pair. And that causes a mutation. If we look again, here's the DNA. The next one. During the replication process, if the cell happened to put an A in place of that C, there would be a mutation in the next generation. A mutation like that then gets translated into a protein. So here is the normal protein, and here is a mutation. So now an amino acid is changed in the final protein from a histidine to a proline. And that could, for example, lead to resistance to a particular antibiotic. As I mentioned, only one change is needed in a ribosome to become resistant to streptomycin. And so it could be that. So that's one source of variation, that DNA isn't always perfect. It also can be damaged, and one of the most famous types of damage is due to UV light, which you can get from sun, for example, which can cause what they call thymine base pairs here. So instead of being linked across, as it should be, they become linked to each other. And that creates a problem in the DNA. When it fixes that problem, it's very likely to put a mistake in there because it doesn't know what the right base pair was. And so that causes mutations. And that's why you should wear sunscreen, so you don't get a lot of mutations. So bacteria are the same. Bacteria can be affected by chemicals or UV, for example, and get mutations. So a single base pair can cause resistance to some antibiotics. These are rare. Typically, it's on the order of 1 in 10 to the 7th or 10 to the 9th cells that get a specific base change. But there are often many, many, many cells. In 1 milliliter of media in the lab, we have 10 to the 9th cells. So we will have a mutant in there.